For years, the cannabis industry has centered its aromatic identity around terpenes. The consensus has held that these aromatic oils are the sole architects of how the plant smells. It is an elegant theory, largely because it works so well for the pleasant profiles. We know exactly how a compound like limonene builds a bright citrus note, or how myrcene constructs the scent of a fresh pine forest. But there is a glaring chemical contradiction in this model. While terpenes explain the fruit and the pine, their chemical structures fail to account for the plant's most aggressive, pungent odors. Think about the infamous strains on the market. Terpenes alone cannot build the sharp smell of raw gasoline, pungent garlic, or startled skunk. Curious if these familiar aromatic oils are chemically incapable of creating those overpowering, room-clearing stenches, we have to find the compound that is actually responsible. It turns out our baseline understanding of these heavy aromas has been incomplete. The true source of the funk requires looking at a completely different branch of chemistry that has remained hidden in plain sight. The actual chemical architects of that deep stench belong to a family of molecules called Volatile Sulfur Compounds, or VSCs. Specifically, cannabis produces a group called prenolated VSCs, like 3-MBT. These sulfur-based compounds are a common botanical defense mechanism. They are the same molecules that give hop cones their sharp, biting bitterness during the brewing process. You can find this chemical family in the kitchen as well. They are the compounds responsible for the sharp, lingering pungency of raw garlic. And, true to the name, these are the identical defensive compounds deployed by the animal known as the skunk. In a volumetric comparison, there's a massive discrepancy. Terpenes are relatively abundant, making up 1% to 2% of a flower's weight. VSCs, however, are just a barely visible speck. Zooming in reveals they exist in microscopic fractions, concentrations measured in parts per billion. The reason they dominate the smell profile is biological. Human olfactory receptors are hyper-tuned to detect sulfur, meaning even a trace amount can instantly overpower an entire room. The intensity of the scent is a result of this extreme sensitivity. Because our receptors are so tuned to sulfur, a microscopic amount of these compounds can overpower a high volume of terpenes. A plant doesn't produce these sulfur compounds by chance. The ability to generate VSCs is a strict genetic prophecy, hard-coded directly into the plant's DNA. During flowering, a biological assembly line kicks into gear. Specific gene clusters within the DNA unwinds to produce specialized enzymes. These enzymes pull raw sulfur from soil nutrients and bind them to precursor molecules to forge the VSCs. These specific gene clusters are highly concentrated in certain lineages. You will consistently find them driving the profiles of legendary strains like OG Kush, Chemdog, and true Afghan landraces. A gassy or skunky profile is the result of a deliberate, genetically programmed extraction of soil nutrients. The next question is how this affects the user. Beyond the aroma, inhaling these intense sulfurous compounds alters the physical experience of the plant. Emerging research suggests that VSCs actively participate in the entourage effect. They interact synergistically with primary cannabinoids like THC and CBD. While we often look to terpenes to steer the mental aspects of a high, VSCs appear to govern the physical response. VSCs anchor the physical feeling that users describe as melting What
was I saying?